Harvard with a postdoc at WashU St. Louis. I met Paul in 2016 from WashU at Dartmouth. Interesting meeting because my very good much regretted James Scott had been the geomicrobiologist at Dartmouth for several years. Here it is years later, and here it's Dartmouth. But also, very good. So we started talking, and so I was going on and on about aerobic hey, what about aerobic? Aerobic. That is the well, it turned out that the chemistry, so here you go. You're going to hear some really good stuff on the clumped isotopes of methane by Leavitt in this crazy organism that doesn't say there, Pseudomonas stutzeri, that lives in the pelagic sea.
Awesome. Thank you, Doug. I'm going to start my timer here so I give myself a 5 minutes heads up to shut up. Thanks all for coming today. I really appreciate you coming out a week after AGU in the what I call a wintry mix in New England. So this is very at-home weather for me. Feels like spring, however, given the cold and damp. But yeah, as Doug said, I'm going to talk to you today primarily about this really interesting system that my recent graduate student has been working on in collaboration with Doug and Ed Young out at UCLA. And I'm going to hopefully save a few minutes at the end to talk about a somewhat, or I should say very different isotope system that we're working in, which is the hydrogen isotope systematics of Archaea lipids as a means of thinking about paleoclimate. But if I run out of time, I'll skip over that. So for the first bit, we're going to talk about unclumping what's known as the methane paradox in the surface ocean, and then dive deep into some clumped isotope systematics, which hopefully you all have been introduced to by this guy here. So as a microbiologist and a geochemist, I like to say I do both, but I don't do them together all the time. We're really challenged with this problem of thinking about essentially the enzyme scale or the microbial scale and how the processes and reactions occurring on the short spatial scale, short temporal scale influence essentially planetary or exoplanetary phenomena. And so really transiting this sort of space-time continuum, as my colleague Fike likes to put it, is really the fundamental challenge, is we're understanding processes in the lab over very short spatial and temporal scales, but we're trying to understand how these can occur on planetary and geological timescales and what those signatures can be. And so I try to basically just break this down into a simple box model problem, because that's sort of the geochemist in me. And so all the microbial processes I like to think about, I just put in this sort of cellular metabolism box, independent of what it actually is. And these are things like growth rate, protein expression, redox state inside the cell, metabolite concentrations. These are sort of the intra-process challenges that we're trying to understand. But these are all subject to the boundary conditions of your given environment. And so these are sort of all your standard states, your temperature, proton activity, electrochemical processes, maybe nutrient limitation if you're worried about how you feed these organisms. But then you're going to get some sort of stable or hopefully geologically stable outputs like lipid structures we'll talk about at the very end. But today mostly we'll talk about organic and inorganic isotope systematics and molecules that hopefully are preserved for some interval of time in the geological record. Or in the case of methane, depending on the process, perhaps actively being generated and enforced in the atmosphere. Another inorganic isotope systematics, and actually as Professor Hazen here has demonstrated, changes in mineral phase over time can actually be a good indicator of changes in not just redox state, but biochemical state. And so sort of thinking about this as sort of the working model, this is essentially how I approach all the problems in my laboratory. So the first vignette, which is really going to take up most of my time today, so don't worry, when I get to part 2, that's actually closer to the end. I sort of fell backwards into this, trying to understand this marine methane paradox, which my graduate student, Lena, did most of the work for. And then we've done a lot of great measurements out in Ed's lab, supported by the DCO, of course, and then now by the Simons Foundation and NASA. But a whole host of collaborators that have helped me with some of the other isotope and microbiology challenges. So the observation that's been around for about 50, 60 years now is that in certain parts of the surface ocean, so the semipelagic, hemipelagic, is you get oxygen saturation and methane saturation in the photic zone. And this seems to be problematic in that oxygen and methane don't really mix well in terms of you're sort of at these two end members of oxidation state for the oxygen and the methane. And as far as we know, all the biological processes that produce methane can only really occur in fully anoxic or completely anaerobic systems. And so for about 50 years from the time this was discovered in the late 60s, early 70s, these observations have sort of plunked up all over the globe in really low nutrients, so really low phosphorus systems. And this was essentially the key that some folks, Dave Carl and Dan Rapetta, used to come to understand this methane maxima, or this methane paradox in oxygenated surface waters. So the way they've sort of pieced this puzzle together, and then what we're going to try to apply stable isotope and clumped isotope systematics to really interpret, is that you can get essentially aerobic bacterial strains in the surface ocean that are under constant phosphate starvation. They need phosphorus and nitrogen to grow, and there's essentially no free inorganic phosphorus available to these microorganisms. And so to get that phosphorus, some organisms synthesize these organophosphorus compounds, or phosphonates, as we'll call them, MPN, I might say a few times. And this tends to get locked up in the DOM, or the dissolved organic matter fraction, that's just constantly being generated in this surface ocean by photosynthesizers. These organisms have what's known as a CP lyase. You can just think of this as the little micro machine, the enzyme that actually attacks that carbon-phosphorus bond. And they're after the phosphorus, but they release methane as a byproduct of this reaction. And so this is essentially the process that we're sort of using as the staging grounds for understanding isotope systematics in a fully oxygenated or aerobic methane generating system. So that's really why I got into this, is to try to understand what are the isotopic signatures of this, and then can we use that isotope signature to look for this process elsewhere in nature. So breaking this down in a little bit more detail, this is essentially a simplified version of the methylphosphonates. These are usually locked up in big honking organic molecules. This is what you can get from Sigma off the shelf. You get this reduced phosphorus bond, which is somewhat rare in nature. Usually you get a phosphorus 5, you get a phosphorus 3 here, bound to this methyl group. And then the CP lyase, so the enzyme that we're interested in, again, just think of this as the mechanism by which this bond is being broken, is actually going to oxidize that phosphate group and generate our methane, our molecule of interest here. And this is what our target population of molecules is when we're going in for both bulk stable isotopes and then eventually clumped isotope analyses. I should just say, one of the great things about working with enzymes that are very well understood is you can actually sort of map out the reaction sequence in somewhat fine detail, because there's really good biochemists like Kamat's group down at UT Austin who actually crystallized not only the enzyme, but then they've done in situ biochemical studies of this. So we actually have a pretty good sense of the fundamental reactions occurring in this system. This is one of these Etiopia diagrams, which I'm not a huge fan of, but I think they're useful for sort of breaking down the delta D, or the delta hydrogen, in SMO space versus delta C13. And these are sort of broadly the populations of where you see biological and abiological processes in the isotope systematic. So there's a relatively large field in abiotic methane generation processes, and I'm just sort of keeping it generic for now as biology versus non-biology. And then you have your canonical sort of anaerobic methanogenesis processes, so primarily microbial, and then he's broken it down by the different types of biochemistry. But really, there's a lot of overlap in these fields, and so really the first question we had was just the generic, where do we fall in isotope space, and then is that actually useful in trying to interpret these surface ocean methane populations or reservoirs. And so that's essentially what really the first arc of a GRL paper that's just coming out is, and I'll just summarize it for you here quickly to get onto the clumped. But so what Lena did for her master's thesis was basically to grow these 5 different strains of surface water microbes. So these are organisms that eat sugar or complex organic matter and release methane just simply as a byproduct of being grown under phosphate or phosphorus starvation. And what we see them do is under these, they produce basically methane as they grow. And so independent of the temperature that they're growing at or independent of the strain, we have a bunch of different strains here, they all produce methane, which is basically just the baseline of what we need. What's interesting is that the carbon isotopic fractionation, so the epsilon or the fractionation between source MPN and methane product, is effectively zero. Sure, you could argue there's a few per mil in there, but it's a very tiny isotope effect for carbon. Carbon can fractionate upwards of 100 per mil. And so this is sort of the first order observation, is that independent of temperature or strain, we see very, very, very small isotope effects, which is really the key observation that that Lena made here, particularly when you put all of her experiments, over 100 different experiments, in perspective. So this is the MPN experiments, with the canonical CO2 reduction methanogenic pathways, or the C1 or the acetate methanogenesis pathways. So these are sort of your canonical anaerobic methanogenesis pathways that we've all discussed in the literature since Whittaker and earlier colleagues. And what Lena discovered is that there's basically no isotope effect between your MPN source and your product methane. And this, if anything, this is the bulk isotope signal of this process, is that there is no isotope effect. And so that was a little bit frustrating at first, and then I realized, well, this is actually useful because now we can directly track the carbon that's contributing to this phosphonate reservoir in natural systems. So in the ocean, we'll just sort of do a little thought experiment. If I can advance the slide. There we go. In the surface water at this site off the coast of Hawaii, where we know it's very, very phosphorus starved, and this has sort of been routinely observed this is the Aloha station, is that you have your canonical sort of oxygen saturation and methane supersaturation in the surface waters, and those concentrations drop with depth. So this is very clearly a surface source process. If you look for copies of that gene, that enzyme, the CP Lyase, the average copy number per genome that a colleague of mine calculated from a bunch of metagenomic data, you can see that it basically peaks right up there in the subphotic zone, so about 350 meters of water depth. So again, this is a surface process. It's not occurring at any depth where you would predict the anaerobic processes. And then isotopically, the methane that's sitting up there is about 40 to 45 per mil depleted with respect to the international standard. And you can see from the concentration curve and the isotope curve, this basically just looks like it's being oxidized and depleted as you go, or enriched, excuse me, as you go down with depth. So really, this is the surface water signal, and it actually reflects very nicely the methane in the atmosphere right over the site. And so basically, we can apply our CP Lyase isotope effect, or fractionation factor, to this, and we can estimate the organic matter phosphonate carbon from just a simple inversion of that effect. And what's really neat is this actually directly matches the observations of that moiety in the surface water that some other folks have published. And so this is really sort of just a nice application of this, but we're hoping that when our colleagues from Woods Hole go back and actually start to do this as a depth profile through this system, we'll start to see if that holds up or not. One of the really interesting caveats to this, and I just point this out because I didn't put it up on the slide, or did I? Nope, is that there's actually two other processes that can operate in the full presence of oxygen. And we don't understand them very well. One is a nitrogenase enzyme. I shouldn't say that can operate in the presence of oxygen, but it can operate in cyanobacteria. Nitrogenase can make methane by accident. And there's another unknown mechanism in cyanobacteria that's still sort of under investigation. But up till now, the CP Lyase is probably the most likely candidate for generating this methane with this isotope signature in the surface ocean. So that's just sort of a nice handy application of what Lena found. But sort of when I got into conversations with Doug about trying to understand this process with respect to other methanogenic pathways, we quickly came back to this diagram from Etiopia, sort of the summary of the different biological and non-biological processes. Oops. And so if we just put it in context, at the Aloha site, we're seeing about a 40 per mil depletion, and in hydrogen isotope space, we see about a 250 per mil depletion. It's very non-diagnostic of a biological, or of any process. And so if we were putting this into a different system, say a soil or a subsurface reservoir, where we really want to try to understand what are the different biological versus abiological contributions, we sort of quickly come into, this is the same type of axes, just reverted. We come into this problem of the abiotic and the biotic processes. They sort of have distinct fields, but there's a huge amount of overlap. And so that these are relatively non-unique bulk isotope signals. And so to hopefully better understand, am I whining? To hopefully better understand these isotope systematics, we really wanted to turn to using the tool of clumped isotopes that Ed and Doug and others have developed over the last 10 years or so. And so I'm just sort of flashing the data initially, but what we want to understand is now this C13H3D clumped isotope metric and the C12H2D2 clumped isotope metric. But we're going to use our really weird process of this aerobic methane formation to try to understand the fact that you get a lot of your abiotic gases and your equilibrium-looking gases close to this sort of theoretical prediction, whereas a lot of your microbiogenic or microbially generated or kinetically generated gases are coming well off this equilibrium distribution or prediction. And so we're going to hopefully use this tool of this metabolism to try to understand why do you get this offset. So that's really what we're after here in this next little section. So some of you may have been at the DCO wrap-up meeting a few months ago, or you've probably seen Ed or myself use this type of slide previously. But basically, when we're thinking about clumped isotope systematics with respect to methane, we're thinking about the rare, what's going on with that whining? We're thinking about the rare bonding of either a deuterium to a C12 or multiple, a deuterium to a C13 or multiple deuteriums to a C12. Those are essentially our major clumped isotope species. And really this comes down to the fundamental bonding properties of that rare isotopolog bound to a rare isotopolog, and you can see that it essentially slows down in this molecular dynamic situation the vibrationals, the row vibrational frequent states. And so really we can take advantage of those properties to try to say something about the formational conditions of these molecules. Now it's fantastic when they're formed under purely equilibrium conditions and where they're actually completely mixing isotopomers, isotopologs amongst the species. And that's a really useful process in that it allows us to then, I'm gonna skip this, allows us to predict something like temperature from that. And so if this was a purely equilibrium distribution, this just becomes a thermometer like a lot of other isotope systems. And so in the case of C13H3D, you can get temperature predictions based on, we'll call it the rare isotopolog abundance that we'll discuss in a moment. And you get an even more sensitive thermometer with the more rare isotope log C12H2D2. And what's fantastic about this is that you can then just take Ed and Edwin's calculations here, and you can just invert those and calculate temperature, basically. And so in principle, this is a beautiful two-axis geothermometer that you're then able to use just by measuring the clumped isotope distributions of these two rare isotopologs in whatever methane population or sample you bring back to the lab. Now, of course, that requires a very large radius isotope mass spectrometer. And so this is the panorama that some of you are very familiar with, sitting out at UCLA and Ed's lab, basically made possible by DCO, so thank you, DCO. And this instrument is really what allows us to make these measurements. In addition to the enabling technology, you need enabling people. And so this has been a great collaboration with Doug, who's really the one that got us involved, with Ed's group, and then Lena Tansar, who is my master's student, is now a PhD student at Woods Hole, and Janelle, an undergrad, and then of course Jabran Labidi, who used to be a postdoc over at UMD. These are the crew that really made these measurements happen. So many thanks to them for hours and hours in front of the vacuum line, and then hours and hours in front of the mass spec. But so to come back to sort of our main problem here is that we get we have this equilibrium, this theoretical prediction for temperature, and some natural gases seem to obey, or seem to at least fall in the realm of that equilibrium distribution. But in this framework, a lot of the microbiogenic, or the microbially generated gases, do not. And so this is really the fundamental question, is why are these looking so disequilibrium? Sure, we know they're kinetically generated, but there's only really two ways we can get this far off of this axis, and it's got nothing to do with your independent, your isotope, your bulk compositions. And so this is sort of the challenge that was put to us. When we first made measurements of the CP Lyase, or the MPN-generated methane, the first observation was that it basically fell way off this distribution, the equilibrium prediction, in clumped isotope space. So this is actually somewhat consistent with what I would have predicted prior to making these measurements. We know it's a kinetically generated gas. What's interesting, though, is when we then started to label the water in which we grow these organisms, and then therefore with which we're putting hydrogen or deuterium onto that methane, it pulled it right back onto that equilibrium distribution. And so this was sort of the first head scratcher when Lena made these measurements. I thought the instrument was broken. It was not broken. I thought we'd fractionated the gas in the vacuum line. We had not. This was a very robust result that we could reproduce. And so this was really the first indication that we had somehow manipulated the clumping of our gases, even though we're generating it in the lab the exact same way, just by varying the water isotope composition out of which we're generating the methane. So this sort of sent us back to trying to understand, in Ed's parlance, or Ed Young's parlance, is really trying to get at the question of what can drive you essentially up and down or vertically on this C12H2D2 axis, but sort of be ignorant or not give you any change in this first order, this first dimension of clumping as C13H3D. And so really this was too large of a gap. We jumped about 60 per mil, or 40 per mil, excuse me, in C12H2D2 space. There's really only two processes that we understand fundamentally that can move you on this axis. The first of which is essentially what's known as a quantum tunneling effect, which Ed and I just like to summarize with essentially this simple one-dimensional energy diagram, excuse me, two-dimensional energy diagram, where rather than having to go over your classic sort of energy surface, the hydrogen atoms are essentially small enough that they can basically, it's not quite burrowing through, but you can essentially ignore this energy barrier that they need. The other option, and this is just a little bit of simple algebra, is that in this isotope space, when you're defining your reference frame, you have to make a few assumptions about the way isotopes sum up. And so really what this comes down to is that we're using either the geometric mean or the arithmetic mean when we're considering our methane isotopologues, when we're defining our clumped isotope axes, which I skipped over quickly. But basically, your axis, rather than an internationally recognized standard, is simply a theoretical framework in which you're operating. The challenge being is that there is this inequality when you're using either the arithmetic versus the geometric mean, where your geometric mean is always going to be lesser than or equal to your arithmetic. And this has a really peculiar challenge when you then when you forget about this, or when you sort of ignore this in clumped isotope space. And I'll try to illustrate this point briefly here. So if you consider two gases that you're going to make clumped isotope measurements on, either molecular oxygen, OO, or carbon monoxide, CO, you have a number of different options for your clumped isotope species. With oxygen, you're always going to be bonding an O to an O, so you basically have your 16, 17, 18 option for either site. Now, those oxygens can be coming from either the same reservoir or two different reservoirs with either the same fractionation factor or different fractionation factors when you're forming that O2 molecule. Now, the same is, of course, true for oxygen and carbon going into your CO molecule, but these are always coming from separate reservoirs. There's no way you can get a C, it's just sort of evident, or self-evident. Certainly, you can have different isotope effects associated with them sourcing from those different reservoirs, but they're never fundamentally coming from the same reservoir. And this is a really critical observation with respect to how you then write out the reference frame in this clumped isotope space. And so the word I haven't used yet, which is what I'm coming to, is that you can basically get what's known as a combinatorial effect associated with the fact that you're pulling oxygens either from the same reservoir or from different reservoirs with different isotope effects. Now, to illustrate this for methane, what we basically see is, now you can sort of just in theoretical space think about the site-specific contrast for two hydrogens coming into that methane molecule, say from, again, the same water reservoir or from the same water reservoir with two different isotope effects or different isotopic reservoirs with different isotopic compositions. And what that does to the relative abundance of your minor isotopolog C12H2D2, where you have these four sites, equal amounts of deuterium or hydrogen in each, is that depending on the difference in your two reservoirs or effects, is you can basically get this, what looks artifactual offset in the relative abundance of these two different sites. And so basically we're really curious about is the isotopic difference that's somewhat illustrated here between those two reservoirs. Whereas for the C13H3D, you can increase or decrease the overall abundance, but the relative difference between those sites is never actually going to be changing because you're always contributing you're always contributing equal proportions from those different reservoirs. So hopefully illustrated and then said slightly differently, turns out this CP Lyase system, or this odd aerobic methane generation system, is a really good way of doing this because we know we're always getting the methyl group coming in from whatever its precursor is. In this case, we just think it's this MPN type molecule. And we're always getting water, one hydrogen or deuterium from the bulk solvent, which in this case is just water. These are being essentially mixed at the enzyme site, and then you're putting this 3-to-1 or 3/4, 1/4 onto that methane. You cannot violate this with this enzyme, and so we always know our stoichiometry, and we always know and we can always calculate our fractionation factors, which is really effective for then trying to understand how we're moving in this clumped isotope space based on that combinatorial effect that I sort of alluding to here. So Lena decided we're going to go do this experiment, and so we basically ranged the water over a huge range in sort of delta D space, so upwards of about 7,000 per mil. I had a bunch of Antarctica lying around my office, so we used some 400 per mil depleted water in delta D. And sure enough, we see a very consistent linear relationship between the methane and the water. This is perfect. This is exactly what we would have predicted. And this is really useful because we can also calculate the fractionation factor between by the enzyme when it's adding that water hydrogen or deuterium. In clumped isotope space, and so now I've switched my two minor isotopolog species, but this actually stacked up with what we predicted. Interestingly enough, is that in the C13H3D, there's basically no effect. We're within the about half per mil standard error that we would get with any normal analysis. And then we see this very interesting nonlinear pseudo-parabolic relationship between the water composition and the C12H2D2 composition. And so the real challenge at this point was to really be able to try to build a model. It's just a simple combinatorial model that allowed us to essentially back this out. Fortunately, Ed and Lena were able to do this with cheerleading from me. And basically what they can do in a very simple combinatorial model that Lena is running in just Fortran is to actually predict these measured values with a very simple set of two reservoirs. So basically your water isotope reservoir and your methyl, or your MPN, carbon and hydrogen isotopic reservoirs. So there's a little bit of scatter. We might be missing a clumped isotope fractionation factor, we think, which would actually capture all of the data. But essentially, with only a two-component, two-fractionation model, we're able to explain the vast majority of the data. So that's really exciting. Now, this is a bit of a weird process, especially for methane isotopolog people, this isotope effect, this CP Lyase. And so really what we're hoping to do now is apply this to more of the canonical methanogenic pathways using CO2 type reduction, where we would predict there's no combinatorial effect because all the hydrogens should be sourcing from water. And then we're going to apply this as well to the C1 and the acetotrophic pathways. So the prediction is we'll see this for some of the pathways, and this is just a very complicated diagram to say exactly this. But basically, when you have C1 or methyl type groups or acetoclastic, because it's basically fermentative methanogenesis, these processes should show us exactly this combinatorial effect when we vary the water in which they grow. And then conversely, the hydrogenotrophic, when you basically start with CO2, you strip it down to bare carbon, and then you build it back out to CH4 from hydrogens sourcing from reservoir water. In principle, we should see no combinatorial effect with CO2 type methanogenesis, but we'll find out. This is sort of the next step. So I went through that a little bit more quickly than I expected, but that's good. So methane from phosphonates basically reflects source carbon. This is useful in just bulk isotope space when you're in an environment like the surface ocean where you have a good handle on what your putative sources are. However, this is obviously more challenging when you move to other natural systems or oil and gas reservoirs where you have a lot of potential mixing or oxidative processes. And so that's where clumped isotope systematics hopefully are starting to help us understand not only formation temperatures in the higher temperature systems, but formational pathways in the lower temperature kinetically dominated systems. And then looking forward, we basically need to sort of take this approach to those really canonical methanogenic systems. as well as some of the abiotic pathways that operate at lower temperatures, so Sabatier type reactions, some of which were done here by Dionysus. So I'm hoping some of those types of experiments can come up. I can convince you guys to do them. Questions thus far? I'm going at lightning breakneck speed. All right. So I'm going to take a step back, and we're basically going to apply this same framework to some of the very recent and very different types of work we're doing on hydrogen isotope systematics in bacterial or microbial lipids, and so large organic molecules. And really, in this instance, we're trying to understand some of the environmental information, in particular, either temperature or different hydrological properties from the lipid structures, but more specifically, the lipid hydrogen isotopic composition of those lipid structures. And so this is essentially a lot of work that my postdoc, Yuki Weber's been doing down in Ian Pearson's lab, and then some fantastic experimental work by Alice Zhao and Beverly Chu, my lab manager and recent graduate, as well as some other folks out at CU Boulder and Seb Kopp's lab where we actually make the isotope measurements. As I was telling somebody earlier, we do all the experiments in my lab, and then we farm out, or I send samples out and then send graduate students to go hang out in fun places like LA or Colorado in the middle of winter. So this is really based, this project's really built on this paleohydrology proxy idea that the plant wax community has developed over the last 20, 30 years. Really actually with a lot of early work that was done here at the Geophysical labs by Marilyn Fogel, Kate Freeman when she was younger, or I should say early in her career, excuse me. And then really trying to understand or trying to invert the observation of the hydrogen isotopic composition of something like an N29 alkane plant wax, basically, and getting something like the hydrogen isotopic composition of meteoric water or precipitation. And what's interesting is that in this review by Saksa, is that there seems to be a reasonable correlation between these two values. And so this has been applied to essentially modern and paleo hydroclimate, just trying to understand, as you move a water mass over a continent or deposit it into a lake or into a glacier or into snow, can you invert some of that isotopic information, not just from the water, which you might not have 100,000 years later, a million years later, but from plant waxes that might be well preserved in some sedimentary basin. And so using this as essentially the inspiration, the microbial geochemical community turned to bacteria, and I'll walk you through these fun diagrams in a moment. But what it turns out is that fractionation between lipid and water in the bacterial world basically was a disaster. And so that's really all I'm trying to show you here is depending on which organism you grew, how you grew it, what you grew it on, the isotope effect is on the order of 600 per mil in both directions. And so this was basically not a very useful paleohydrology proxy if you can't really tell what your original water was, because the composition of the magnitude of fractionation is varying completely independent of the water itself. So our goal was basically to take this framework and march into the other domain, the Archaea, which synthesize very diagnostic lipids, so the structures we are very comfortable with, we know who makes them, or we have a good understanding of who makes them. And then we also have a good understanding of the biochemistry, so we understand the vectors of the inputs of hydrogen into ultimately that lipid hydrogen. And these all more or less source from energy carriers like NADPH. All we really care about is that hydride that contributes about half the water onto the lipids, hydrogen. And then potentially a quarter of the hydrogen coming directly from water and potentially a quarter from other metabolic intermediates. But we have a really sort of simple or I should say very basic understanding, fundamental understanding of how this actually works in theory, both in bacteria and Archaea. Now we want to go apply it to some archaeal organisms that actually produce these lipids that are very diagnostic of environment. And so we're going to do this with a thermoacidophile that we grow in my lab. And just to sort of give away the punchline, and then we'll try to understand it, it gives you about a 200 per mil isotope effect. And then a surface ocean dwelling Archaea in Meritmus gives you about a 270 per mil isotope effect. One of the really interesting, at least hypotheses about Archaea is that they're just phenomenal at existing in low energy environments. And this is a hypothesis laid down by Dave Valentine about 10, 12 years ago now, is that if you sort of survey all of pH temperature space, you tend to find the Archaea, not exclusively, but for the most part, Archaea tend to dominate the polyextremophilic, or sort of the multiple extremes of the natural system of nature where we do find life. So essentially, the lower pH systems at higher temperature or the higher pH systems at higher temperature. What this really means for them is that they have to find a way to protect the inside of the cell, or sort of inside operations, from this really caustic outside environment. And one of the most efficient ways to do that is to build a really rigid, really robust membrane. But these membranes also have to maintain a certain level of porosity and permeability such that they don't become too impermeable to, say, nutrients or other energetic sources. So this is really the game that these organisms have to play, in that they have to be able to alter the saturation and the structural composition of their membranes instantaneously or very quickly to respond to assaults from the natural environment. And so really this is one of the amazing inventions of these archaea, is these isoprenoid type lipids that I've cartoonishly drawn here. And they essentially often have these monolayer membranes of 40 carbon chains decorated with up to 8 of these cyclopentyl and sometimes cyclohexyl rings. And so these are just known as GDGTs, or we'll just call them archaeal isoprenoids for today, so we don't have to get into acronyms. But these are very diagnostic lipids of the archaea that we're interested in, both from the surface ocean as well as from these hot spring environments. So if you find these in a marine sediment or in a hot spring sediment, you have a pretty good sense of who the organisms were that made them, relatively speaking. So we're hoping to then use our understanding of hydrogen isotope systematics and try to understand, and our understanding of who synthesizes these lipids, to then ask questions about the water isotopic composition of that environment. And so the first part of this was really developed by Yuki, which was this basically just a methods development, which took him only a mere 2 years, but to basically get these biphytones, which is the core lipid, out without generating an isotope effect, or doing it in a way where you're always labeling it as you go. And then being able to separate them very cleanly on an LCMS to be able to make continuous flow isotope ratio measurements on then a GCMS at high temperature. And so basically, these are just the 0 ring, 1 ring, 2 ring, 3 ring of those cyclopentane ring biphytanes, as we call them. And then one of the really unique ones is this 2-ringed cyclopentyl, 1-ringed cyclohexyl, the BP3', that only surface ocean archaea make, as far as we know. And so these are really diagnostic lipids for these organisms in particular. And then these other ringed structures are somewhat more common. You can find them in hot springs as well as surface ocean archaea. But they're also very diagnostic of just archaea in general. So we have a good, like I said, we have a good sense of who's making these. Now, in terms of understanding the isotope systematics, we do a bunch of very constrained growth experiments in my lab, as well as down in Ann's lab. So we're basically growing these in continuous cultures at the various temperature pH regimes that we're interested in, harvesting the lipids, and then going through Yuki's very complex process to get out a hydrogen isotope measurement of each biphytanol, or each core lipid. And so basically 2.5, 3 years of work by a number of postdocs and wonderful graduate students, Yuki and my grad students. We find this really interesting systematic or pattern. So in the surface ocean archaea, you always see this patterning of the biphytanol 0, 1, 2, 3, where it's always increasing, always becoming more enriched with ring number. But there's basically no response to growth rate. And so we can grow them at steady state across a very large range in growth rate by limiting them for their electron donor. And we see essentially no variance, which was a little bit surprising at first. This is a bit more what we were predicting from the earlier work in bacteria, is that in our sulfolobus, our hypothermophile acidophile, you do see a consistent, we see that same patterning of the biphytanol 0 through 3, but you see it, the mean is shifting with growth rate. So basically when they're growing really, really fast, which is the lower values here, they're more fractionated. And when they're growing really, really slowly, on the order of about 2 days doubling time, you start to approach essentially a plateau, or excuse me, the magnitude of fractionation decreases. And then you can just basically break this down by how many measurements we've made of each individual biphytanol. We don't have to worry about that for today. The nice thing about these organisms is in both cases we know the biochemistry really, really well, which I won't bore you with. But the value in that is that we can just develop a simple isotope flux balance model from that. And so if we want to understand the isotopic composition of our biphytanol as a function of water and the other various growth parameters, the big contributor, as I said earlier, this gives about 50% of your hydrogen is sort of coming through this NADPH intermediate. The key being the fractionation factor associated with the transfer between these two molecules is on the order of about 1,000 per mil, depending on which set of experiments you believe from the literature. So you have essentially a huge lever of fractionation factor here, of magnitude here. And so understanding if and how this varies as a function of growth condition relative to water composition, this is what's going to allow us to understand the output, or the fractionation between lipid and water in both these cases, and hopefully get us back to originally water, which is really what we're after. So we can do this, or no, we've done some other simple modeling to try to understand the patterning, which seems very consistent. I won't bore you with that right now. But really what we're after here is that depending on how you write your flux balance model, or excuse me, depending on how you write it, depending on how you parameterize your flux balance model, depending on the assigned value for your fractionation factor of this transhydrogenase, as it's called, and the flux, you're really getting about 80 to 90% of the hydrogens onto those biphytanes through this pathway. And what's really interesting is that this is true independent of whether or not you're growing as an autotroph, which one of these strains does, so fixing CO2, or a heterotroph, which just means they're eating sugar, breathing oxygen. So the really nice thing is we can basically reproduce this patterning with a very simple flux balance model. This is just to show us that basically this negative pattern is essentially never predicted independent of our parameterizations. And really what we're seeing here, and this is probably the simplest way to sum it up, is we're basically seeing a pseudo-equilibrium effect between NADPH and NADH. And we're basically seeing how we have the relative consumption of that NADPH pool. So basically a Rayleigh distillation of this reservoir of hydride as it's being put onto that lipid. And we think that's essentially what's controlling either the flat pattern in the really slow-growing autotroph versus the faster-growing heterotroph. But there is an end-member value in both cases, and these slow-growing autotrophs seem to be operating basically at that end-member fractionation, and that fractionation factor is hopefully giving us a value that we can then invert for environmental water. There's a little bit more complexity to the aerobic heterotroph, but we can deal with that. So this is essentially going away from the cartoon and trying to show us, and just showing us that basically our end-member calculations for that fractionation factor are about 600 per mil in both cases at set flux values of about 90% in each case, and we get really good fits to both these data sets. Now, we can't distinguish between this and a fractionation factor that's larger in the case of the autotroph because we basically get no slope, but this is really the only consistent outcome that we can get for the autotroph, excuse me, the heterotroph. And so if this holds in the autotroph, we seem to have a pretty good understanding of the system as a whole at this point, but we still need to do a few more experiments to demonstrate that. So this is really, that's the lab work that we're still churning through. My postdoc gave a version of this talk last week at AGU just to sort of set up the fact that we now need to go finish understanding the systematics, but now we actually need to go out into natural systems and harvest as much of this really diagnostic lipid, Archaea, as we can get, and see if this actually shows us trends in precipitation or hydroclimate. And so we just sort of speculating a couple weeks ago, or actually last week, I guess, some of the best places we think to do this would probably be really deep lakes. Obviously, the ocean's isotopic composition does not vary terribly on short time scales, so 100,000 to million year time scales. So we've sort of just put together, or I guess Powers put together a really nice map of all the putative places you'd find Archaea, which is that lipid I was just talking about. And now we're arguing that we should probably get some money and go sample these lakes, certainly in the sediment core, as well as in the surface waters, to try to get enough of this lipid out to then make these measurements, either as a function of depth inverted for time, or over seasons if you're working on the surface waters. Lake Lagugno is a lake that Yuki's worked a lot on in Switzerland, and this is just a really nice example where we know you have a lot of these Archaea that produce that compound. So that's the name of them, Thamarchaea. They make a lot of this Archaea. It's very consistent with the environmental parameters that he's measured in terms of electron donor and oxygen availability. So we have some sites. Now we got to get out there and actually do the work. Other applications, Ann and I were kicking around other ideas as to how to put this to value to not just paleoclimate folks, but potentially thinking about sort of indigenous versus exogenous, or allochthonous versus autochthonous Archaea, and really starting to think about the dual isotope systematics of the carbons. You have 40 carbons approximately on each of these chains. So you can start to get at carbon source as well as hopefully something about hydrogen isotopic composition of the precursor water. So these are really sort of, the door is hopefully open now for folks that are interested in this type of measurement. And so we'll have to see where it goes. But this is pretty much, this is the latest as of really last week, 2 weeks ago. So we have a lot of work yet to do in the hydrogen isotope space. Yeah, so I've said all that, but sort of bring it all back together. The clumped isotope systematics and bulk isotope systematics of the methane have really helped us a lot in understanding not only our fun little process in the surface ocean, but we're hoping this process of aerobic methanogenesis will help us understand clumped isotope systematics broadly writ for low temperature processes. And hydrogen isotope systematics in Archaea, I walked into that realm very skeptical because the bacterial lipids were kind of a disaster during my postdoc. But we're actually seeing that there is potential as a paleoclimate, or at least as a hydroclimate proxy. So we'll see where that goes. But thank you very much. I tried to keep the biochemistry to a minimum. Oh, sorry. Yep. Absolutely, yeah. And so this is that. The best answer to that is we don't know those exchange rates very well. There's a nice review by Alex Sessions, I think, a couple of years ago, where I want to say he showed, or he sort of pulled out of the probably literature that folks here are more familiar with than I, is that depending on clay content and temperature is sort of what are going to really dictate a lot of that exchange frequency, or rate, excuse me. And so, yeah, there's going to be a finite limit to how hot and how deep and how long you can go with hydrogen isotopes and biomolecules. Absolutely. I do not pretend like that's not a thing. So really, I think targeting younger and colder environments is going to be critical. So we don't get too lost in the diagenesis weeds, which is always the fun processes. But yeah. Absolutely. Absolutely. And so I think, yes. So the nice thing about some of these lipid structures is that they are very diagnostic. And so once they get to a certain point in their structure, a better organic geochemist than I can say that has been essentially partially reoxidized. And then hopefully we can ignore it based on that or sort of invert that. That said, the carbon isotopic composition, I'm hoping will be able to give us a sense of whether or not this has been partially not at all, or just what degree of partial oxidation or reworking, as you're saying. But I think this is also going to be part of the challenge, is to work carefully through that process. Also, not just looking at the structures and the lipids, but also what type of environment are you in. Bring a good sedimentologist with you is what I always like to say. Don't go to high energy environments. Yeah. 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 I'm trying to think where that would be most valuable. Certainly during the biosynthesis of them, I think it would be really interesting, and you also have a lot more control because you're in the lab. But like certainly with like the acetate molecule, which is one of the major precursors, I think Kate Freeman showed this sort of using bulk isotope geochemistry through a lot of challenging, basically degradation processes. That's an aside, but yeah, thinking about in situ measurements of a carbon, hydrogen, multiple carbons, multiple hydrogens. That would help us at the moment in trying to understand basically where does the hydrogen come from during biosynthesis. We have this sort of 50, 25, 25 estimate, or 50, 75, 25, depending on how you write your model and whether or not you're growing an autotroph or a heterotroph. But that would, I think, be very helpful, the multiple isotope measurements simultaneously, in actually understanding do these, like, when is the water actually being transferred onto the lipid? Because that is actually, we write our nice little box models, but that's very a gross simplification of the biochemistry. So I'm sort of punting on your question because I actually don't know if you could do those types of they're using an Orbitrap. I don't know if those types of measurements would work on a 40 carbon compound at this point. I know they're working on like serine at this point, so we'll see. Yeah. Okay.
which is very useful.
Mm-hmm. Okay. Mm-hmm. Okay. Okay. So you were working at, or they were working at really high deuterium contents in the water. Okay. So there is an observation, at least I vaguely remember this, maybe this experiment was done here, but upwards of about 20 atom percent deuterium. Basically, you can just map growth rate onto atom percent deuterium in the water. And it, like you said, it diminishes, and so there's clearly a cost. But it's interesting that they're dumping it into the lipids as opposed to somewhere else. Yeah. Yeah.
It's the safest place.
The least reactive moiety that you have to deal with. Yeah. Yeah. Right. No, that's okay. That's interesting. Yeah. Kinesis. So to be honest, we did not grow them, we did not sacrifice them carefully enough to distinguish that. I would be surprised if there was one, but the short answer is we don't know yet, so we haven't looked. That said, we did grow them, or Lena grew them and sacrificed them all at late log, early stationary. Actually, honestly, stationary in all cases, and that was mostly just to let them grow out, so that we weren't hitting a moving target, so such that you would do in like a normal batch experiment where you're sampling, with a certain T and sulfur isotope systematics, like I did for my PhD. You see huge, well, I shouldn't say huge, but tens of per mil variation in bulk isotope space, depending on whether or not you're in early log, mid log, or stationary phase. So rate certainly matters in bulk isotope space. With clumped isotopes, as far as I can see, we haven't seen a big systematic with growth rate broadly, but I don't think anybody's done the experiment carefully enough yet. Yeah. Your bulk isotope effect will definitely change, and that we can actually probably look back at our bulk data. I tend to not look at the bulk data very carefully. You should see a fractionation factor there. Actually, not with our process, but if you looked at like a normal methanogenic process, you would see it, because our process has no real isotope effect, or sub 1 per mil isotope effect. I wouldn't try to interpret what we've seen in the bulk isotopes, but for CO2 reduction, for example, or acetoclastic methanogenesis, or C1, those are actually experiments I really want to do, which is another reason we run chemostats in my lab, is trying to do, in particular, the C1 metabolism over, say, 5 growth rates of, say, a 12-hour doubling time to a, say, 100-hour doubling time, if we could hit it, and then capture the bulk, the methane. In principle, you should see, I'd guess, about a 20 to 30 per mil isotope effect difference in bulk. But the clumped, well, that's the question at this point. Is there any difference in function of rate? Yeah. Sorry, I tangented. Thank you all for coming on a snowy day. Yeah.